Hi guys, today we're going to start our first lesson in our cell growth and reproduction chapter. So this is part 4 of our cells chapter. This will be the last part that we go over before we move on to some genetics. So this is going to be kind of the base of all of the stuff that we're going to have to know to fully understand how genetics works, because genetics starts with how cells grow and reproduce. So we're going to start today with an overview of what cell division is and why we have cell division in the first place. So all cells come from pre-existing cells. We know this from the cell theory that we learned about at the very beginning of the year. Okay. Cell division is the process by which we get new cells from those pre-existing cells. Okay. Now there's different kinds of cell division. It's different for prokaryotes, which are those bacteria cells, those very simple, small organisms. And eukaryotes use cell division and go through cell division in a very different way. So eukaryotes would be things like protists, fungi, plants, animals. Our cells are eukaryotic cells, so we use a different kind of cell division. Prokaryotic cells use cell division primarily to reproduce. That's kind of the only way that they are able to make more of themselves, whereas eukaryotes are able to reproduce in other ways. So prokaryotic cells have to actually split themselves into 2 new cells in order to produce more of them. Okay, so that's why they would use cell division. Now, eukaryotic organisms can sometimes use cell division to reproduce. So that would be any kind of unicellular eukaryotes. So one single-celled organism that's a eukaryote would use cell division to reproduce, because that would be the only way that they could. So that would be things like those protists are really the only eukaryotes that we see that are unicellular. Now, for all the others, like fungi, plants, and animal cells, they use cell division to grow. So when organisms grow, they make more cells, and also to repair tissues. So like if you get a cut or something like that, your skin cells need to produce more skin cells to help heal that cut and close that up. So you have to make more skin to cover up that space where it was damaged. So those are the primary uses of cell division for those different kinds of organisms. Now, here's some reasons that we might want a cell to divide. Primarily for growth, and that's mostly for multicellular organisms. So when organisms grow, the cells don't get bigger, okay? They multiply, they get more cells. So the number of cells that you had as a baby are not the same as the number of cells that you have now. Another reason we might want to use cell division is for repair and replacement of cells. So again, this one applies to multicellular organisms. So it would be organisms like us and like plants. So if something gets damaged, we need to divide and make more cells in order to repair that thing. And then lastly, we would probably use a cell division for the reproduction of the species. So this would be mostly unicellular organisms, some multicellular too. It just depends on the type of organism. So especially for bacteria, they have to divide into multiple cells in order for them to be more of them. They can't reproduce the same way that we do. So those are the main reasons that we would use cell division. Now, with cell growth, okay, there are some limits to how big cells can grow. So the first 2 things we talked about there had to do with cells dividing. Now we're going to talk about how cells can grow, because some cells can get bigger in a sense, but they have a limit to how big they can be. So in general, most living things are going to grow by producing more cells, not by increasing in cell size. So like I said earlier, you, as a baby, the amount of cells that you had as a little infant is not the same amount of cells that you have now. You have definitely more cells than you used to. So you increase the number of cells that you have in your body in order to grow. Your cells don't get bigger. Okay. Now, the reason for that is that cells are limited in their size by 2 major factors. Okay. One of those is the surface area to volume ratio, and the other is what we call DNA overload. So we're going to go into detail with what these two things are here. So first we're going to take a look at the surface area to volume ratio. Okay. So what this is the idea that cell volume increases at a faster rate than the cell's surface area. Okay. So basically what that means is if the cell gets too big, it can't receive the amount of food and oxygen it needs to support the entire insides of the cell through the cell membrane. Another thing is that it can also allow for waste to build up and can actually poison the cell. Okay, so this little illustration kind of helps us to understand that. Okay, so here we have a surface area of just 6 millimeters, okay, or square millimeters, I should say, on the outside of this little cube. Okay, now the volume of this cube, though, is 1 cubic millimeter. Okay, so this little purple thing inside of the cube is the cell, let's say, okay, the inside of the cell, and the cube itself is the outside of the cell. So we have a very small volume, but a nice big surface area. So that big surface area is going to allow us to receive more food and more oxygen than the volume really needs. Okay, and it's going to be easy for this cell to release the poison because it doesn't have a whole bunch of distance to cover in order to get out of there because the volume is small. So this is like ideal conditions for the cell. They don't have a lot of space to cover and a lot of space to require nutrients for, so it's going to be a lot easier for that cell to survive. Now, if we were to increase the surface area to 24 square millimeters, that also increases our volume to 8 cubic millimeters. But notice our surface area jumped way up from 6 to 24, whereas our volume only went from 1 to 8. Okay, so the surface area goes up at a much higher rate than the volume of the cell does. Now that's a problem because, as you see as we get to this big one, see all that empty space on the inside? Okay. There's a lot of ground to cover to try and get the food and oxygen and nutrients that the cell is going to need to the important parts of the cell. So that's going to take a lot of energy, and the cell can't produce enough energy to do that. Also, there could be a lot of waste products building up on the inside here, and the cell doesn't have enough energy to get them all the way out here. Okay, so that's why we can't have a gigantic cell. That's why all cells are super small. So that is one of the main limiting factors to cell growth. Okay. Now, our other limiting factor is DNA overload. So DNA overload is starts with the idea that the nucleus contains instructions for making proteins, which we know that is DNA. Okay. DNA is our instruction booklet for how to do what a cell needs to do. Now, those proteins, or enzymes, are used by almost all organelles to carry out different cell functions. Okay. Now, the amount of DNA in this cell does not change regardless of cell size. So, if a cell gets too large, there's not enough protein to fill the cell's increasing requirement for that protein. So this kind of goes to an information crisis, we call. So the cell is kind of overloaded, overworked, trying to produce enough protein that it needs, but it can't because it's too big and it can't make enough. So then it kind of goes into this crisis mode. I kind of imagine it like that scene in SpongeBob where they're like, he's the working at the restaurant, and like the only thing he knows is fine dining and breathing, and you got all the little SpongeBobs running around trying to find his name because he can't remember his name. That's kind of what would happen in a cell in this case. If it gets too big, it can't get enough instructions to make enough proteins for what that cell needs, so they'd be running around like, where's the recipe for that protein? Okay. So if it gets too big, you're going to have the same amount of DNA no matter what, but you're not going to be able to make enough protein for that cell to properly function. Okay. So those are the two main reasons that cells cannot grow to be gigantic things that we can see without a microscope. Now, one of the first steps in cell division is copying DNA. Okay. So since those instructions for making cell parts are encoded in DNA, every single new cell we make must get a complete set of DNA. So this is what we're going to get into in our next chapter with genetics a little bit more. So this requires that the DNA be copied or replicated before a cell divides. That's one of the most important steps because you're not going to have a functional cell if you don't have DNA inside of it. So basically what happens is your original strand of DNA is going to split into two, and then the cell is going to go through special processes that we'll go over later that match up these different bases and make two new strands of the DNA. Okay, so that is the most important first step of cell division. Now with prokaryotes, cell division looks a little bit different. Okay. Prokaryotes divide into two identical new cells by the process of binary fission. So binary fission is when you take one parent cell and that parent cell itself divides into two new cells. This is what we call a form of asexual reproduction because there's only one parent cell. Now, the important thing to understand here is that they're identical cells, okay? They are not two genetically different cells. They are basically clones of each other, okay? So in binary fission, the chromosomes or the DNA that are attached to the cell membrane, so what they do first is they attach to the cell membrane and they make a copy of itself, and then the cell grows to about twice its normal size, and then a cell wall is going to form between those chromosomes that have attached to the cell wall on opposite sides. So then that parent cell that has grown to twice its normal size is going to kind of split in half into two new identical daughter cells. So that's what we call it parent cell is what you start with, daughter cell is what you end with. Okay. So important here, they are identical. They are clones of each other. So this is what that looks like. You have that prokaryotic cell, that parent cell, makes a copy of its DNA. Okay. So now we have two little rings here. Those little rings attach to the cell wall or cell membrane in two different spots. And then you can see that the cell kind of begins to divide. You see those little divots here, okay? Then eventually that cell is going to split into two identical daughter cells, okay? Now, cell division in eukaryotes is not quite the same as prokaryotes. Okay, eukaryotes have all of those different organelles inside of them that they have to account for, right? Okay, they have a nucleus and all of those membrane-bound organelles like a mitochondria, endoplasmic reticulum, all that stuff. All of those organelles have to be copied exactly the same, so the two new cells that form from division will be exactly alike. So there's a lot more work that goes into cell division in eukaryotes. The original parent cell and two new daughter cells also have to have identical chromosomes, just like we saw with the prokaryotic cells. So because of this, DNA is copied in different, there's different phases to the cell cycle, which we're going to be going over here in a bit. So the DNA is copied in what we call the S phase of the cell cycle, and organelles found in the cytoplasm then are copied in the growth phases. So that kind of organizes the cell division and allows for these things to take place at separate times so then the cell doesn't get overworked. So both the nucleus and the cytoplasm must be divided during cell division in eukaryotes. So the nucleus is divided in what we call mitosis. That's when the genetic information is divided. And then the cytoplasm and the rest of everything else is divided during a stage that we call cytokinesis. So you can kind of remember that the cyto prefix goes with the cytoplasm. So cytokinesis is when the cytoplasm is divided and cell division is officially done. Okay. That's where we're going to stop for this lesson. For today, we'll talk about the cell cycle in our next lesson.